Hey everyone, so I am going to do another flipped classroom today, and I am doing it on cellular transport. Now, if you're taking notes in your notebook or obviously on a Google Doc, I would say make this a whole new topic, a whole new Google Doc, because now we are getting into another subcategory. So we just talked about, we did viruses and bacteria, we did prokaryotes and eukaryotes, and we did cell organelles. Now we're doing what's called cellular transport. So we're going to just talk about that today, and we're only going to be talking about, I guess I'll get into it and then I'll explain what we're going to do. So the biggest thing with cellular transport is that every cell needs it. You need it for homeostasis. Now remember this wonderful, wonderful, wonderful word, homeostasis. It means to keep everything equal. It means to keep everything in balance. And that's what cells want. That's what your body wants. Everything wants to be at homeostasis. So how this happens is that cells need to move materials in and out of the cytoplasm in order to make sure that this homeostasis keeps track. And there are a couple ways that this happens. So there's passive transport, which we're going to be talking about today, and that's all we're going to be talking about. This is the only thing you are going to be getting quizzed on is passive transport. There are three types, and you do not need energy for this. You have diffusion, you have facilitated diffusion, and you have osmosis. Then there's active transport, which uses energy, and again, you are not going to be quizzed on this today, or on Friday. So we're only talking about passive transport, and that's the only one you're going to be quizzed on. All right, diffusion. Diffusion is pretty simple. It literally just means moving particles from an area that has a high concentration of it to a low concentration. And again, there's no energy needed for this. And we say that we're going down the concentration gradient. So if you actually look down at this picture right here, you see that there's a high concentration of these really like blue dots. And as we move down the concentration gradient, there's less of them. So we're going from a high concentration to a low concentration. So think of it this way. Here we are on a super microscopic level. Let's say that the red dots were like a food dye that food coloring dye that I put into a bunch of water. Let's say the orange dots are water. I put it into one section, and then what eventually happens out is that through diffusion, the food dye is going to go throughout the entire thing. They don't just stay in a localized area, they get spread out. So that's really what diffusion means. Done. Okay. I just want to talk about some terms that you need to know for our next thing we're going to talk about. So there's polar. So if you have a polar, I guess I should actually say polar molecule would be better, and like a non-polar molecule. I think that makes a little bit more sense. So polar molecules have a slight charge to them. Examples are like a chlorine ion, hydroxide. We have ammonium, which is NH4+. Water is actually polar. It doesn't have a charge, so it doesn't have that negative positive next to it. That's because it's slightly positive and slightly negative. So it actually does have a charge to it. Where there are non-polar molecules like CO2, doesn't have any charge to it whatsoever, so carbon dioxide. CH4, which is methane, we talked about that with the archaeobacteria, that methane gas that they release. That doesn't have a charge to it either, so that's considered nonpolar. Okay. Diffusion does occur across the cell membrane. Woo! This is because it is a lipid bilayer. Remember we've talked about this, that phospholipid bilayer. This is a cell membrane. That's exactly what it is. So we have that two layers right here. You can see it. We have two layers here, and they are made of mostly lipids, hence the lipid bilayer. So cells need a variety of things to come in and out of the cell. They need ions like chlorine and sodium. They need small molecules like oxygen and CO2, and they need glucose, which is also just a fancy name for saying sugar, which is C6H12O6. It's a long dish. And then certain molecules can go across this lipid bilayer, and then certain molecules can't. This is why we call the cell membrane a selective permeable membrane. Or I'm actually going to just change this up, and we're going to just make this our semi-permeable membrane. Some things come in, some things come out. It's as simple as that. All right. This is where I want you to know about polar and nonpolar. This is why I talked about it earlier. So some small polar charged molecules can actually pass through the lipid bilayer, but larger molecules actually need to go through what we call these membrane transport proteins. We talked about these on Monday, so I guess today. We talked about these earlier you guys went around the room and you were getting, the notes. That's what this is. That transporter proteins are is so important because if something is too big, it can't go through without this transport protein. Even though we need it, you need to have a transport protein, to make sure it goes across. Now, that's where facilitated diffusion comes in. If something is too large and it needs a transport protein, that's when we call it facilitated diffusion. It just means, think of the word facilitated. Facilitated literally means to help out. So if you are being facilitated by something, you're getting help some way, somehow. Facilitated diffusion. You need something to help get you across the membrane. So again, these are really large molecules. These are proteins, carbohydrates, things that are way too big that can't get across without them. Okay, cool. All right, osmosis is next. Osmosis is the diffusion of water. Just water. Nothing else. Just water. Makes you realize that. I'm being obnoxious about it because diffusion is everything else. Osmosis, water. So again, movement of water. This is very important for keeping homeostasis. Remember, water is an extremely important substance. We cannot live without water at all. We have about, I think it's like 3 days. If we don't have water within 3 days, we can die. So they actually go through, because they are polar, they go through a channel protein that is called aquaporins. Kind of cool word. But when we are doing osmosis, the same thing as diffusion, we're moving from a high water concentration to a low again. So high to low. Now there is no energy needed in this. At all, actually. So that's pretty cool. So there is absolutely no energy needed. However, again, that's because we're going from a high to a low concentration. Anything that's passive transport, going from high to low, no energy. Osmosis is one of those. Okay, cool. We're rocking this. All right. 3 types of let me rephrase that. There are 3 types of osmosis: hypotonic, isotonic, and hypertonic. This is just going to talk about the certain types of solutions where the high is versus the low. It's actually going to affect cells greatly. And so we're going to talk about that. So first we're going to talk about hypotonic. This means the amount of water is greater on the outside than in the inside. So my example right now is in an animal cell, and I'm just going to talk about blood cells. Blood cells are very reactive to water. They're very sensitive. So what I mean by that is that they will have these drastic effects if they get too much water or too less water. So we're going to talk about that. So that's why I'm only talking about blood cells right now. I'm going to talk about plant cells in a little bit, but right now just animal blood cells are a really good example. So like I said before, hypotonic means that there is too much water outside of the cell than inside the cell. So this involves, since there's too much outside, that means that all the water is going to move inside because there's a higher concentration outside, and we always want to go from higher to low, down the concentration gradient. Whew. So, animal cells do not have cell wall. Only plant cells do. So our blood cells don't have a cell wall. So when there's too much water that goes into them, they actually swell and they get to a point of having so much water. Think of a water balloon with too much water. What normally happens if you have a water balloon with too much water? It bursts. It's the same exact thing that's happening here. When you have a water balloon that has too much water, it will burst. Same with a blood cell. If it has too much water, it's going to burst. If the water does not get removed. And that's just if there's too much water in the surrounding area. So, yeah, there you go. All right. Isotonic. Isotonic is like the perfect balance. So we want to be in isotonic all the time. That's because water is equal outside and inside the cell. So there's no movement of water in or out because it's imbalance. And actually, just a little cool fun fact, 0.9% solution of saline, so salt water, is actually isotonic to animal cells. So what does that mean about our blood? Just think about that. We're going to talk about that. So I guess I'll put that a little bit down here. Explain what the above point means. So kind of an interesting thing. Just want you to think about it because it's pretty interesting, that 0.9%. And we'll talk about it tomorrow. But next thing, hypertonic. So we went hypo, iso, hyper. So hypertonic, this means that there is a greater amount of water inside the cell than the outside. So then the water wants to go again. It's always trying to get to isotonic. It's always trying to get to balance. So then the water is trying to go outside as fast, as much as possible. So that's the big thing. So when that happens, the cell will shrink as the water keeps flowing out of it. And then when a cell loses water, it actually can't function properly, and then it can't divide, so more can't come from it. So this is a big deal that this kind of happens. You don't want this to happen, neither do you want them bursting. So pretty much at all times, you always want that nice isotonic, perfect balance. Lesson! Okay, we just talked about animal cells with our blood cells. Now we're going to talk about plant cells. Plant cells react very differently. Now remember when I was talking about hypertonic, or did I just mess all these up? No, okay. Hypotonic. Remember when we talked about hypotonic when like it burst? Now that was because it doesn't have a cell wall. So plants have a cell wall. They want to be hypotonic. So if you see right here, this is like turgid, which means that it's normal. That's when like a plant, like you can see my plant behind me right here. This is opposite. This plant behind me, it wants a lot of water. So if a plant is standing up nice and straight, that actually is it's normal, like what what it wants to be in. So that's really good. Isotonic for an animal cell is where we want to be. However, in a plant cell, that actually means it's flaccid. So it's kind of like, that's when we see like a plant kind of drooping and things like that. And then lastly, when we have our hypertonic situation, this is not good for a plant cell. This is when it's like, it's shriveling up and dying at this point. There's kind of like, if you actually look, you can see like everything just gets sucked in and it starts to shrink and everything comes off of the cell wall. So you have that rigid, nice cell wall still, but everything's shriveled up inside of it. So here's my question for you. How would the vacuole have a big part in this? Why would a big, large vacuole in a plant cell, why would this matter? And why does the vacuole such a big part of this, of keeping, that hypertonic, isotonic, and hypotonic? So that's my last question for you. Well, that was pretty awesome, guys. You guys did a good job. Remember, you can go back, you can pause, whatever it may be, and we're gonna go over all of this tomorrow in much greater detail. And the big thing you need to realize right now is that all this stuff, you do not need energy. You're going from a high concentration to a low concentration in every single one of these situations. So, good job today. I will see you all tomorrow. Be good, be kind, and I will see you all very soon.